anyway, World Antimicrobial Resistance Awareness Week. It's my pleasure to wrap up the festivities of this week by introducing my good friend and colleague, Dr. Eric Nelson from the University of Florida. So Eric and I met online as Eric sent me an email in 2017 or so, or maybe before then, because he read a paper that I wrote that he liked, and I think we cited one of his papers. So I was a bit aware of his work as well, and it started as just a conversation by email and some Zoom calls that turned into some collaborations. I'm happy that those have really gone in all kinds of interesting directions since. So in the interest of time, I won't belabor the background that Eric has a background in microbial ecology. He did a master's in microbial evolution and ecology, and went on some cruises doing aquatic microbial ecology before moving into more medical or clinical domains. So he did an MD and got interested in phages and cholera, so working with Andy Canoli, and then through fellowship at Stanford, and then eventually to the independent group to Florida. So Eric is going to talk about one part of his research program. He does all kinds of interesting other work, very applied clinical stuff in Haiti. He has a Canadian context today. It's one of the parts of the right. So anyway, it's been a pleasure having Eric here over the week. Some of you met him on Wednesday at the symposium. It's my pleasure to introduce Eric and hear a little bit. So join me in welcoming Eric.
Today's title is Defending Against What If There Were Two Pups on the Ice? This is the work of many, many people, and this is a super collaborative and fun field because you get to be the expert on maybe your one little wedge, but then there's many other wedges that you're not the expert on. You have to be very gracious and humble to have fun collaborations to make these big projects work. And so some of the teams that I'll be highlighting today come from Emory, from the International Center for Viral Disease Research in Bangladesh, here at McGill, colleagues at Hopkins, at Stanford. And so it's a really big team. And the body of work is over at least 10 or 12 years. And so there's been many funders through this journey, including the Canadian Institute for Health Research. So thank you to CHRI. And then also in terms of investigators, there's Sharia Ahmed at Emory, Ishay, who's a graduate student here today, Saeed, who's a former graduate student of mine, Naima, who you know quite well, Patrick, you haven't been to the McGill server farm, and you want to know where the Shapiro server stack is, you look for the disco stack, because it's the only stack that's producing light and is making disco colors. So it's been really fun meeting everyone and getting to know everyone quite well. I don't have any conflicts of interest today. I will say that we just started a not-for-profit called Mango, which is what we're calling a global health technology partner. Take a lot of software we developed and graduated out of academia to improve clinical decision support. So apparently, on November 1st, just a year ago, the Wild and the Lightning were playing the game, and puck number 1 got shot up in the nets. And what happened was got stuck there, and they thought the puck was gone, and the referee threw a new puck on the ice, and then this one came down, and the keeper was looking at this one, and Rosie picked it up and just snuck it around the post. And what I want you to think about today is that puck number 1 is antibiotics. You know, we're really kind of stuck in this mindset of drug exposure, and then we study rates of drug resistance emergence. But what I want to kind of put into your head today is that there may well be a second puck on the ice, and it's this interaction with phages and phage resistance. And so I get tongue-tied around the use of antimicrobials because I'm trying to put actually phages into that bucket, and it's something that we're trying to get our minds around. It's something that I'm hopeful that I can bring into the conversation. So in broad strokes, the mission of our lab historically has been to think about how better to respond to large-scale infectious disease outbreaks. we do that through the lens of cholera because it's really a model system for that. And what we do is we think about in that journey, from the patient's perspective, all the different ways that we mess up. So I worked in Port-au-Prince in 2010 and Zimbabwe in 2008 and Nepal, all these different places around the world. And I see these common themes of ways that we mess up. And so one of the things that makes our lab different is our kind of perspective and approach to this challenge. And so the approach that we take is to think about to improve outbreak response. We first need to consider the totality of the challenges faced by the patient from the patient's perspective. And as I've kind of gone on in my career, I thought about that quite a bit and what I do professionally. And actually, if I take a step back to think about our group, what we really do as a group is advocate for human rights. And we use our trades of medicine and science to give voice to marginalized populations because it's a marginalized population that suffer the bulk of complications in events like cholera outbreaks. So that is the reason why we push, why we stay up all night running GPUs with Google to make some data for this morning, and it's where we find motivation. So in broad strokes, what this perspective and approach looks like is goes on this following timeline, where so some of you may have children or loved ones that might fall sick at night. If one of Jesse's kids falls sick at 2 in the morning night, there's this question of how sick is my child? If they're sick, do I seek care? And from whom do I seek care? So in Haiti, over the last 10 years, the NIH has been funding our group to think about how to extend care early to families to avert morbidity and mortality. And so this is manifested in something called MotoMeds. It's a telemedicine and medication delivery service that goes online every night at 6 and goes till 5 in the morning. And we motorcycle meds and or nurses out to households overnight before children get into a pre-emergent state. This is really important for cholera because you have a competition of your favorite pathogen. Cholera may be the #1 in terms of time of exposure and time of death, and hours really matter. And the benefit of cholera is that unlike many other diseases, something as simple as oral rehydration solution or Pedialyte can take a mortality rate of 20% and put it down to 0.1 or 0.01%. So I was not trained in this, but if I want to reduce morbidity and mortality, it's important. If a family does seek care at a clinic or a hospital, there's a really important series of decisions that have to be made. The first is, from the clinician's point of view, how sick is the child? We've done things in the WHO, green, yellow, or red. And the process and the guidelines for how that's done is surprisingly not that evidence-based. So we work on a fair amount of modeling and clinical decision support tool development to help that process. The next question is, like many of the clinical people in the group face, is when to use a diagnostic, and then if you use it, how do you interpret that result in the context you're working in? So we also work on that question, and that starts to get to the question of once you have a triage level and a diagnosis, for diarrheal disease, it doesn't matter what the agent is. The maneuver is going to be resuscitation, and that can be orally, it can be IV, and then you have to figure out what those amounts are. So we also work in those domains. Now, something that we were talking about at ASTMH last week and on Wednesday is this question of what to do not with the antibiotics when the bacteria are resistant to the drug, but what to do if you're in the case of cholera, where you have a cholera strain that's sensitive to azithro, doxy, or ciprofloxacin. Those pan-sensitive. What guidance is there to decide which of those 3 drugs to use? And in our situation, we'll make recommendations at nation or continent levels. And so if there's different rates of AMR emergence, we would want to have those data to inform which drug we use. So that is another domain. And then I mentioned the WHO. So there's a group called the Global Task Force for Cholera Control. I have the pleasure of being on the clinical and the laboratory group of that agency. And basically, it's a joy because I can take these buckets of research and convey them to the WHO, and they can say, great, Eric, thank you, but we also have this other thing that we're struggling with in Malawi. And then they'll send that to me, and then we'll start working on it. So today we're going to focus on more the microbiology side, but I think it's really important to kind of put this in context. So today we're going to talk about a problem, a need, an approach, and an outcome. So I mentioned a little bit about the problem already, which is that antibiotics being puck number one, and phage being puck number two, impact cholera in known and unknown ways. And there's really a need for a new framework to investigate how multi-agent interactions impact, in this case, human disease. And I know that there's a colleague, maybe on the internet, who studies oysters and phages and vibrios. And so this is not specific to humans. We need a new framework for this. And then in that framework, we need a really powerful analytical pipeline to assess these agents at scale, because we struggle a lot in the analytics on sample size and having balanced sample sizes. And then for outcomes, I'm committed to three ambitious outcomes. One is microbial evolution, disease severity, and diagnostic performance. And I'll challenge people in this audience that if your favorite disease is UTIs, or bacterial meningitis, or bacterial sepsis, a lot of the principles that we'll talk about today, I think, lend to your favorite disease. So we're going to get into the problem. And this is, for the epidemiologists, what's called an SIRS model. H are new entrants into a system. S is susceptibles. And then in the cholera space, you can have an incredible gradient of patients that are symptomatic, I, or asymptomatic. If you're in an endemic situation like Bangladesh, serological surveillance studies suggest you might have one symptomatic case and 1,500 asymptomatic cases. If you were with me in northern Zimbabwe, 2009, 2008, it was an immunologically naive population, and everyone was symptomatic. I mentioned a little bit about the mortality rate, that's little m, and then with ORS, and sometimes IV solution, you can enter the recovery pool, and then in time, as a function of the durability of the immune response, you can re-enter the susceptible pool. Cholera will exit for the human in a hyperinfectious state. We think about Shigella as having a very low ID50, but actually cholera from a human can be 15 or 20 cells. Coming out of a test tube, it might be 1 million or 10 million. And that's the reason why it is incredibly rare for anyone in cholera lab to get sick, unless they're working with sample from human. About half of the patients that I work with in Bangladesh will shed a bacteriophage, either virulent bacteriophage of 3 flavors, ICB1, 2, and 3. And then if you're working in a situation with very collapsed infrastructure, let's say Port-au-Prince, there's a short loop where patients can rapidly get infected, or cholera can go into a much more complex environmental stage where it can be culturable or not culturable. So this was in a Nature review from 2009, and we left out one really important thing in this model. It's puck number one. So what we began realizing is that we would run clinical studies where we said, we'd ask the patient, "Did you take an antibiotic?" And they'd say no, and so we'd enroll them. And then in a couple of pilot studies, we started being concerned that the patients did in fact have an antibiotic. So then we did a truth serum study where we collected urine and stool from the patients. We asked them, did they take an antibiotic? And then we looked at how this correlated. And what we found was that about half of the people said they had drug, they had taken drug, and basically every single patient had at least one drug. Most of them had 2 or more drugs, and they're meaningful concentrations. And this is really important because we later found that this rapid diagnostic test, which is a kind of dipstick test that we use in outbreak response, if you have antibiotics, azithromycin, for example, or these virulent phages, the odds of RDT test positivity go down by 80% or more when you have these agents at play. So you can really go down the wrong path, either clinically or epidemiologically, impacted by these particles. Um, Speaking to UTIs, we're talking about writing a grant to ask similar questions and urinary tract infections. So in terms of mental models, I always need a roadmap because I get lost. And a roadmap like this is pretty classical, in which you have some degree of exposure to a pathogen. And then if you do not take antibiotic and the bacterium is sensitive, so if we have grabbed a mouse, you may end up being severe. If the patient takes an antibiotic and the bacteria are not resistant to it, the patient may end up in no dehydration. If they take the antibiotic and the bacteria has some degree of resistance to it, maybe there's a leftward walk from red to yellow. So this is kind of a classical mindset. Now, it's a mind bender to now thinking about a patient co-ingesting a virulent phage. And the way that I walk through this is I go down this path on the right, where a patient takes a drug, the bacteria are sensitive to that drug, and it doesn't really matter about the phage or phage resistance. The patient may well end up on the green side of this. If they don't take a drug, and the bacteria are resistant to the phage, bacteria go about their business, and the patient may end up being severe. If the bacteria does not have phage resistance, there might be a rightward walk over to yellow. And then the super complicated one is the middle one, where you take the drug, the bugs are drug resistant, and the bugs are also phage resistant. You may well end up in the yellow space, and then if the bacteria lack the phage resistance, there might be a rightward walk over to green. So this is a much more complex mental model to think about something as simple as diarrheal diseases, but we're going to wander into this because these are, at this point, largely hypotheses, and we're starting to kind of address branches of this puzzle. Okay, so how would we test this? Because you could test this in animals, but its generalizability would be in question. So let's talk about the Bay of Bengal, the country of Bangladesh. I lived in Bangladesh for a period of time. It's a place I know super well. And it's a diverse country, just like many other countries. And so what we decided to do back in 2016, 2017 was set up a cluster randomized control trial across the country in which our primary clinical endpoint was providing one set of hospitals clinical decision support for food resuscitation, assessment, and antibiotic usage, and look at volumes of resuscitation, antibiotic choice. And then in the background, collect samples from across this network and look at some of the microbiologic outcomes. So we had 2,500 patients in the study from across the sites shown in orange. We did collect samples from the one in the middle, but that was not a trial site. That's our main central laboratory, and we did do rapid diagnostic tests at that site. But all the other sites got a rapid diagnostic test. We did culture, and then because the DNA, in my opinion, is very fragile in some of these samples, we put it in a preservative, the ethanol, the RNA later. Those samples that were culture positive went into our pipeline. Those were sample negative, but PCR positive for virus, for the phages, or for cholera went into the pipeline. And this pipeline is expensive, but it's super important. It begins with mass spec to quantify antibiotic. We send samples up to Jesse. He sends them through what I call the Oreo cookies for metagenomic sequencing. We have an AMR phenotyping plate reader that's been running for years that runs AMR profiles anaerobically and aerobically because cholera sees, yes, a rapid diagnostic test. Thanks for asking that question. And then Jesse does the AMR genotyping. So the Chuan back there has a recent paper in which kind of laying the landscape of what's happening in a one-year cycle in Bangladesh. There is diversity at play, even at the lineage level across Bangladesh, with a little bit more kind of diversity down in the southern part of the country, a little bit less diversity up in the north. But in the snapshot of this clinical trial, we actually had competing lineages. From the truth serum work, on the leftward side is mass spec. This is what people say, and we're targeting metronidazole, ciprofloxacin, hydroxy, and aldehydic acid. For those people the know about malabasic acid, it's not super used clinically, and I'm super curious to know if it's coming through the agriculture source. But we do pick it up. It's a kind of an older quinolone, not fluoroquinolone. And so you'll see in blue, these are infections, and then each row is a patient. And you'll just see more blue here versus here. And when I was training, there was this competition between Harvard, Mass General, and Tufts on whose microarrays were the best. And everyone was doing the same study, and they're getting different results. And in retrospect, they were probably all confounded by drug exposures like this. So this is the landscape of the sample type that we work with. And now pivoting over to outcomes, what I want to talk about is kind of a high-level overview of some of the work that Jesse and I have done up until just recently. And I'm going to start with the cartoon that Jesse drew, which is this is kind of the result that I'm showing first, but I think it's important to have kind of the framework to get complicated. The hypothesis originally was that a patient with virulent bacteriophages, simply the detection of that, would be inversely associated with disease severity. That turned out to be wrong. And when we thought about predator-prey relationships between phages and bacteria, when we started thinking about the ratio, we started to make discoveries. And this is analyses largely driven by Naima, who hopefully is on Zoom. So we had this idea of this concept called effective predation, in which you have a high number of viruses, the bacteria, and in the analysis, that correlated with mild dehydration. Inversely, if the predation was ineffective, that correlated with patients having severe dehydration. So there's a lot of caveats, and you have recurrence limitations. And Naima did some sensitivity analyses and found that there's a really interesting threshold that one phage particle with 5 bacteria was a threshold that could differentiate patients with and without dehydration. Now, on an evolutionary standpoint, if you think about the red queen hypothesis, in which pathogens and prey would have to compete with each other and kind of stay in the same place, that hypothesis was tested. And what was found is that if the predation by the virus on the bacteria was effective, the squeeze evolutionarily was on the bacteria, then there's increased radiation of genetic diversity amongst the bacteria. Conversely, if the bacteria had a phage resistance, they could kind of push back against the viral particles. Naima discovered that there was a signature for increased genetic diversity in the viruses. And that hypothesis, it's from Van Halen, it's Van Valen, and from the 70s. But if you look back since the 70s, there's not a whole lot of actually really sound proofs of concept in this kind of infectious disease human biology space, and so it was a nice discovery. So I remember the very first Zoom call that we had when Jesse did all the metagenomics data, and then we slapped on all the clinical metadata, and he had this plot called an RDA plot. And what this plot is every patient is represented by a circle, and they either have blue, mild, yellow, moderate, red, severe dehydration, and then vectors are drawn with factors that are kind of dominating the directionality of how the data is distributed. And so to begin, you can start with cholera in high abundance. So you have the degree of dehydration, you have the clinical metadata, and then you have the metagenomic data also in this plot. And this is about stargazing. It's about hypothesis building. And so we can begin with high cholera abundance correlates with severe dehydration. Side opposite is mild dehydration. That correlates with infection of azithromycin by mass spec. And then if you wander into vectors for viruses like this one, ICP1, you'd expect if our hypothesis was correct that that vector would be swinging over towards mild dehydration, but it doesn't. It kind of wanders in the middle. And there's a reason for that, which we're going to get to on the next slide. So focusing on dehydration, these are box plots, and granted, they're squished towards the bottom, but this is the ratio of virus to pathogen, and this ratio is statistically different than the ratio over for severe dehydration. So one of the things that came out of this was a limitation around the squishedness of these boxes, which Ishay's testing in a whole new different clinical study. But then also, does it matter, this ratio? How does it relate to other factors? So NIMA, in a recent publication that's up on medRxives, put this ratio against other known factors like age and relative amounts of cholera, and found that this ratio for mild dehydration was quite important in being a determinant of dehydration status. Okay, so going on to a second thing, which is why does that vector wander in the middle for the viral particles? And this was kind of one of these things that's, the answer is right in front of us, we just didn't think enough, which is if you plot, in this case, the cases that had azithromycin and model the amount of drug against the relative amounts of cholera, the more drug you have, the less bug you have. And then if you do a subsequent plot of the amount of viral particle against the amount of phage particle, and then you look at a dose response, there's this beautiful quadratic relationship that emerges. And it's clearly has elements of predator prey behind it, and then dose response. And one of the, give you guys all the secrets. One of the limitations with this work, I apologize, is that what these patients are enrolled in hospital admission, so they have a story before they come in, and they have different durations of illnesses and different exposures. So there's actually more kind of dimensionality to these data. So now pivoting in a dangerous way for me over to evolutionary biology, I'm going to present the slides that Jesse and Nyman built, in which to get at how we made some of the conclusions around genetic diversity and evolution, the first step that they did was they looked at increasing amounts of viral particles. And then they binned cholera into a lot of cholera, an average amount of cholera, and a little bit of cholera. And they found that when you had this kind of relationship between a little bit more cholera and viral particles, more viral particles, you had increase in single nucleotide variants. And it took me a while to figure this out, but what I think was happening is that you have to have a significant amount of predation happening to kind of see the signal, that genetic radiation. The next thing that they looked at was increasing abundance of virus, and then the number of non-synonymous mutations in the cholera. And in this case, we had different types of cholera in the study. And so this one that's called ICE minus, that's a mobilizable element that has a phage resistance factor. When you had cholera without the phage resistance with increasing amounts of virus, you had an increase of non-synonymous mutations. When you had phage resistance factors in 5 and in 6, which are strains with this ICE factor, with the phage resistance, you don't see that effect. And then lastly, there's another way of looking at these data. We have non-synonymous mutations in the cholera, and then you look at strains that don't have the ICE element, so don't have the phage resistance, and those with and without the phages, you see that the non-synonymous mutation rate goes up in those with the virus. And then some controls are shown on the right. What am I doing, Jesse?
Yeah.
All right. So it's 12:36. So I'm going to motor through a few diagnostic slides. And I know this is AMR week, it's not diagnostic week. But I think it's really important because if you're going to run clinical trials, you want to account for these questions as potential confounders, make sure you know the effect sizes that you want when you're doing your power calculations. So we've been studying cholera, scientifically for over 100 years, and we're still struggling with how we make the diagnosis. So these are positive results from the same study I just mentioned. This is the rapid diagnostic test, And then this is actually a picture of it. We have a control line, you have a test line. It's just like the COVID test. It's a monoclonal for the cholera and LPS. If you see a band, it's detecting that LPS. This is TCPA, so that's qPCR for cholera. This is culture, and then we have PhD 1, 2, and 3 detected by qPCR. In an ideal world, these circles will all overlap, but they clearly don't, which is a problem that I think people don't want to face, but we need to. So one of the things about the RDT is a lot of diagnostics I think are judged on the limited detection, but another way to think about that is pulling from the ID50 side of things. To think about diagnostic dose 50, in which what's the concentration of bacteria you need for that diagnostic to work 50% of the time. And so in this situation, there's a super relevant dose response where when you're at about 10 to the 8th bacterial particles per mil, about half the RDTs work. And there's a multi-million-dollar deployment of RDTs right now around the world, but they sometimes work, sometimes don't work. And we're trying to figure out if phage particles play a role in that. And if we plot by qPCR cholera against viral particles, and those that don't do a ton of qPCR, the lower the number, the more target you have, and the higher the number, the less target you have. And then we have thresholds drawn these dotted lines, which above that threshold is negative. To the right of this threshold is negative. And so this is the box in which you're positive for virus and bug. This is the box in which you're positive for bug but not virus. And this is when you're positive for the virus but not the bug. So it's a bit complicated. But what I've done is I've put the percentage of RDT positives in each quadrant. So 50%, 47%, 16%, and down here, 10%. So what we're learning is that we really, really need to consider these viral particles not only as confounders, but potentially in boxes like this one as surrogates or proxies for pathogen detection when we can't identify the pathogen we're looking for. This is for ICP3. I'm just going to skip through that because it's the same. And then for those that don't think about 2x2 boxes all day long, this is a 2x2 box where we look at RDT positivity, negativity, and then true positive and true negative. I'm not telling you what true positive is because it's actually a debate. But sensitivity, as you may recall, is A over A plus C, and specificity is D over D plus B. And then we also work in odds ratios as well. So we'll walk you through a bunch of tables. Hang with me. You can make it through. But I'll just show you what we're wrangling with. So the first is, if we look at our population as a whole, how is our sensitivity forming in this test? We have 46%. If we exclude the patients that had phages, the sensitivity actually doesn't fall that much or at all. If we look at the patients that had cholera or ICP1 or ICP3, in that situation, we have a slight decrease in sensitivity. If we look at patients that have mild, moderate, severe disease, and look at sensitivity, we go from 0.25 to 0.43 and 0.63. So if I was an investor in trying to make the world a better place, would I put my money on an RDT that has 25% sensitivity? No. Would I do it at 0.63? I don't know, maybe. And the decision to deploy this RDT was done before this paper was created. And I'm not kind of bagging on the RDT. I'm just trying to provide a context for maybe how we can better use it. So one idea to kind of drill down before we make a recommendation is to kind of get back to that idea of odds ratio to look at phage effects. And so if we bin patients, cholera patients that are phage positive or phage negative, and then stratify them up by mild, moderate, severe disease, we find that with a case definition of cholera detection alone, so cholera by culture, cholera by PCR, we don't see much of an effect between patients with and without phages. But if we expand the definition of cholera to include phage detection, we see, in essence, a 63% decrease in the odds of test positivity, which is super meaningful. To bring this part of the presentation back together, what we're doing in this line of work is saying, at the bedside, we could maybe make a few maneuvers to inform the use of RDT. In the current definition of cholera, being culture and qPCR, we could say, let's restrict the use of patients with severe dehydration, and also ones that say they don't take drug. In that situation, the sensitivity goes from 0.46 up to 0.69. If we expand the definition to include phages, we go from 0.46 up down to 0.38. If we do the restricted mode over those that have severe dehydration and say they don't take drug, we go up to 0.57. So it's a super nuanced maneuver. But we're not actually solving problems. We're just saying, what's the problem that we have? And what's a maneuver you can do for more epidemiologically, maybe use your test more significantly. So it would be a crime if I didn't show the next slide, which Ishay generated since yesterday, which was ranking factors that impact RDT performance, because it's one of these things that make a finding, but then you also want to put in the context. And so just the very beginnings of this, we're starting to do some random forest work, maybe some cat boost work, to kind of rank what are the factors that influence RDT usage, and then move toward making a decision support tool to guide patients at the doctor's bedside. Okay, so that's a lot, right? It is 12:44. So what I'm showing here is the same branch diagram that I had before, and now I'm adding on new hypotheses around diagnostic performance and also disease severity. And I have this one in green down here because these are actually bacteria that come out of the patient and maybe find the next host within a few hours. And you can have this rapid cycling in evolutionary biology happening. You can have diagnostics that are being messed up. And then you can also have maybe shifts in disease severity all happening in relatively short orders of time. Okay, so this is where we pull out our cards. And I gave you some lead-ins on limitations with this work. So, as I mentioned, people are brave enough to hold their card and share some limitations with the presentation so far. You can win an unfunded trip to Florida to go sailing. And if you don't bring your own boat shoes, you can borrow Jesse's. So we can take up a couple minutes because there's a lot going on here, and we are definitely imperfect with our work. So I'm just curious if anyone can bring forward a limitation with some of those science. Yes, sir.
So,
Do you mind sharing your name?
Dario,
So long. Nice to meet you.
yeah. So, my question is about the reporting, the self-reporting of the antibiotic consumption. So, in that self-reporting, you said like when you're reporting to say blood test or urine consumption to the person, probably not working. Do you do the self-reporting before telling them that you are going to take that, or after that you're going to take your time, and how that influences their answer?
Yeah, so the question for the owl, it's called the owl, back there is, what is the risk of primary and secondary gain when I'm asking the question of did you take the antibiotic? I think that's a super fair question. And we do a bunch of different maneuvers to reduce that risk. One of which is that we don't have clinicians or nurses ask the questions. We have a team of researchers that are kind of trained to avoid risk of secondary gain. But it's on the table. The second thing is the word antibiotic is really complicated. There's vitamins, there's supplements, there's antibiotics. antibiotics, there's antimicrobials, there's medications, and that's a super complex question. The other thing is, that was a clinician, and, and she knows that you might have a patient with a respiratory infection last week. They took azithromycin, and then they come in this week, and you say, "Oh, did you take an antibiotic for this illness?" And they'll say, "No, not for diarrheal disease," but they won't think back to the azithromycin. And the azithromycin traps itself in fat super well, and you can leach it for a super long time. So it might not be the patient lying. It's just that they took it for a different purpose, or they just ate some fish or chicken that's just packed full of an antibiotic. Another question. That's a good one. You get a trip before. Another one?
Can I do a follow-up to that?
Depends. Depends if someone else is brave enough to raise the card. You guys are all smart. Maybe one more question. Anything? All right. We'll go from the superior group. Yeah.
I'm on a cholera team, so I feel like I have a solo fair one, I guess. It's just cholera being an acute disease. All the predator-prey kind of interactions you're seeing, are these not, I guess, heavily dependent on the timing of your sample? Like even in the case with you have mild disease and high phage. I would assume in 10 hours that would still be mild disease, but maybe not low phage. And I guess I'm just wondering like how standardized the timing of these samples is, which is, I'm assuming is not at all, but.
So you definitely get a free trip, not free, unpaid trip. Because we're about to go there. What is your question?
So, the follow-up was, how do you explain the self-reported case where they say, I did take an antibiotic, but you have not made a detection with when you analyze the blood report or the urine sample?
Yeah, so the question is, you have false positives with a self-report. Well, yeah, and that's pretty unusual. So when we do like modeling for clinical decision making, if someone volunteers an exposure, in this case, it's useful. And so we value the positive predictive value in that aspect. We kind of ignore the other aspects. Okay, I'll go forward unless there anybody else has a burning question. I can open it up to a non-trainee. Yeah, Ben.
So, kind of brought this discussion of detecting positives and true positives, but the whole discussion is we're kind of working to find a method to have an accurate positive if you don't really trust any of the diagnostic methods. How do we know what the true positive is? I know you had a question about that is a debate right now. But
Yeah, so that's a great question, and I have a colleague, Jason Andrews at Stanford, does a lot of Bayesian class modeling to accept that no one diagnostic is perfect. So can you basically model and then sort of go reference standard that then you can compare the real world diagnostics to? And that's we've gone in that direction once before, and we're getting ready to go back down that path. Okay. I will share with you my limitations. The single time point is probably the biggest flaw with a lot of the work that we've done, because we're making a lot of generalizations around the patient's experience. Sample size. It does require 2,500 patients, maybe even 5,000 to do this work well, and it is super expensive, and the logistics are insane. And so we've spent years trying to get the limited sample sizes we have. And I think that's one of the reasons why I advocate for studying diarrheal disease, because you can get those sample sizes. To study these here in Montreal would be really, really, I think, quite difficult. But we can show proofs of concept in diarrheal disease and then bring the mindset into Canadian or US-based environment to then start testing those hypotheses and make the studies part better designed. Clinical exams are variable. The newer studies we've been running, we've been putting decision support tools at the bedside to get better clinical exams. We can't afford to do mass spec on everything. And so we have to make some assumptions on which ones we use it on, which ones we don't. We're not doing single cell sequencing. So when there's clonality of the lineage expanding in the patient, we're not following that in our current mode. And then Jesse has been very keen on making discoveries around hypermutators. And we haven't really discussed that too much today. Okay, so let's talk about next steps in the last couple of minutes. Through the Welcome Trust, we had this opportunity to run a time series study. And I had an R1 rejected 7 times. And I'm super stubborn. But thanks to those from the NIH and thanks to the Welcome Trust, we were able to run a little bit of that R1 proposal in which we had patients come into the main hospital in Bangladesh. And at the time of enrollment, we captured a stool sample. We captured their clinical history. And then for protocol, these are mainly moderate severe patients. They all got hit with a single dose of doxycycline. And then at 4 hours and 24 hours, we collect samples. And then just like I described before, we could take these samples and put them into this pretty laborious pipeline of first screening for the pathogens that they have, those that are pathogen positive. We can do AMR profiling that doesn't have to be cholera related. It can be cholera plus Shigella or Salmonella or whatever else. We do mass spec for antibiotic. And then we can do metagenomic sequencing. And then we're working with Tim O'Pinen down at Harvard to do some single cell sequencing. And so what we're, the types of questions that we're going to be asking moving forward is across this gradient of pre and post antibiotic, knowing that approximately half of our patients will not have phages, we can ask questions around disease severity, diagnostic performance, AMR emergence, either at the population or the lineage level, prophage induction, which Ben is working on, and then other questions around pathogen and phage evolution. So when the things that keep me up at night, since this is AMR week, is that there are times where I see very limited gains with antibiotic stewardship. And so what I'm worried about is if phages are playing a role in selecting for antibiotic resistance, we need to know more about the phage biology. And that's a picture to kind of share with you the hypothesis. So if you have a population in which you have phage and antibiotic resistance in red, the blue one is antibiotic resistant, and the yellow one is phage resistance, and you have no selection, pressure, no drug, the population will just do its thing. If you have antibiotic exposure, you could get co-selection in red, or selection for blue being antibiotic resistance. If you have phage, you can get co-selection for both, or just selection for the yellows. And then if you expose to both antibiotic and phages, you might have a red dominate the picture. And this is assuming no interaction between phage and antibiotics. If there's a fitness burden with these factors and you remove the selective pressure, there may well be a return to the population norms. And that's another super important question that I hope people will ask. So now what happens if the population starts naive and they acquire this giant SXT element, that ICE element, that has antibiotic resistance and phage resistance? In that situation, without selection, you might have a few events, and then they might backslide over time. They're just blue or just yellow, or maybe it's red, but the rest will kind of stay gray. If you have an antibiotic or phage exposure, you then have this largely red. And then if the interactions between the phage and the antibiotic are antagonistic, you might get a few less red ones. And then if they're synergistic, you might have fixation in the population. So, lo and behold, Jesse Shapiro came to our lab, and we did this in 96 world plates. And it was fun to actually have two PIs doing the experiment. And we did this experiment where we had cholera exposed to doxycycline alone at a slightly higher concentration, and then we had another set where we added phage particles, and then the population with both was able to escape, and then rose up, and then crashed out. And so I would call that this was kind of an element of escape, and then almost like a synergistic ablation. And then at a slightly lower dose, we had this thing where a slightly different concentration phage associated with synergistic distillation without features of escape. So this population with both antimicrobials just calmed down, whereas over time the bacteria with doxycycline just kind of slowly escaped. So what we're trying to do is we're trying to go to patients and with this new clinical study ask these same questions. And these will be the last couple slides. We're almost done. These are the Euler diagrams I showed before. So these are patients and those tests that were positive. In this case, we have a patient with moderate severe dehydration. So we don't see necessarily the lack of overlap that we saw before because there's been a selection bias on the enrollment. But as you go to 24 and 24 hours, we start to see that the circles don't overlap as well. And then we just look at the in like a cross-sectional time series way, the rates of culture positivity, 45, 42, 16. They were only doing nucleic acid extractions in RBTs. Those samples are liquid, so there's a different denominator in these three, but they're going roughly kind of 40s from T0, 40s at T4 hours, and then kind of in the teens and low 20s at 24 hours. So even though we don't have a control arm in this, there's a clear selective event with the doxycycline. And because we have patients with and without phage particles, we'll be able to ask these evolutionary questions. And then going back to CT values, you'll see that at the starting point, we had roughly a CT value of 24 and 26. And then end of the study at 24 hours, our CT values were about 16-fold lower viral particles or bacterial particles. So I think that for just these metagenomic work, there's been enough selection that they're going to be able to ask and accommodate for some of the limitations that we've talked about. Okay, so I talked about a problem, a need, and an approach, and I'm going to hop forward to my last slide right here, which is like the future. So I think that there's a future in which BioFire's qPCR-based diagnostics in the clinical practice start to go away. And we start to incorporate metagenomics to detect pathogen or phage proxy for pathogen. We can do some forecasting and based on the metagenomic profile, is there a drug in that system? And then we can do some risk assessment around severity now, severity in the future, AMR emergence in the future, AMR now, as well as phage emergence in the future, and phage resistance now. Okay, it's 1 o'clock. I finished on time. Thank you.